Okay, Heroes to Hive students. So this lecture is What's in a Hive? And the idea behind this lecture is to kind of give you an introduction into what you can expect to find when you start going through a beehive. This is not an introduction to inspection, so we'll be talking about that in a completely different lecture about the details of our inspections. But this is just a general idea of what to expect to find in a hive and what those parts are. So those of you that are beginning beekeepers that have never been inside of a beehive, this will give you a good indication about where things are located, what you can expect, and where you can expect to find those things. So really, the first thing that you need to be thinking about that you should be doing as a beekeeper, and really that you have to be able to do even before you get started, is you really need to understand that your responsibility as a beekeeper is to be able to identify the problems, the pests, and the diseases that are present in your hive. You need to understand that you have to recognize individuals in the hive and the patterns and the needs of the hive. You also need to understand what you're looking at, what you're seeing, and what that means in terms of the colony as an overall colony. And then lastly, you've got to be able to react to issues with the appropriate measures to ensure that you have a good, stable colony that is in good health all season long. And if you can't do these things, if you can't identify the problems, if you can't recognize what you're looking at, if you don't understand what you're seeing when you're looking in a hive, and if you don't have the ability to critically think and take the knowledge that you're being given and distill that into something that you can use to react to issues that you find, you're going to be in a really tough spot as a beekeeper. That's just plain and simple. And that's why I say all the time, it's so important for individuals that are interested in getting into beekeeping to start the education first. That's why I tell you, if you're a first-year beekeeper, this is not the year to have bees. This is the education year, because if you have never kept bees before, you don't know how to do any of these things. And so it's really important you understand that education is the foundation to success as we start moving forward. Now, this is not just bees. And the thing I want you to think about when we're talking about honeybees is that honeybees are managed livestock. They are not native to North America. They were introduced into the United States, and they are managed just like any other livestock, just like hogs, just like cattle, just like sheep, just like alpacas, whatever you want to talk about as far as livestock is concerned. We manage them the same way. Now, what if I told you that we had a hog farmer that couldn't tell something as simple as what sex their hogs were? That is not the recipe for success. That is going to be a very unsuccessful hog farmer. But oftentimes in the beekeeping industry, I run into people all the time who can't tell the difference between a drone, a worker, or a queen. The two basic sexes and three basic castes, they have no idea what they look like. How can you expect to be a good beekeeper? How can you expect to keep bees alive if you can't tell the difference between a male and a female of the livestock that you're keep caring for? It's the same thing with a hog farmer. And it goes the same with pests and pathogens too. So if you had a cattle rancher that had no idea what pests are going to be problems, what pathogens are going to be issues, do you think that cattle farmer is going to be very successful? Likely no. That the flies alone that attack livestock could really cause some issues. And if you had no idea that stable fly was going to be an issue for your cattle, well, you're going to be in for some rude awakenings when it comes to actually raising cattle. And so it's the same thing with honeybees. If you don't know the pests and you don't know the pathogens to expect and what they can have the impact of what impact they can have on your colony, you are going to be in a world of hurt when those things actually come and hit you. So you have to be able to understand that there is a foundational knowledge that needs to take place with beekeeping. And there is no beginning level to this knowledge. Absolutely no beginning level. It's not like, oh, I can start keeping bees and I'll learn this on the fly. That is not the case. And I have been keeping bees now for 13 years, 13 seasons. And professionally, when I am working with individuals, it stuns me how often I'm talking to someone who calls themselves a 4 or 5 year beekeeper and has no basic foundational knowledge as to identifying the problems, recognizing what's in the hive, understanding how to work within those situations, and then really being able to critically think and take the knowledge needed to make the smart decisions when you get your back against the wall. And really what you have to understand is that all the decisions that happen in your apiary, everything you do is going to be shaped on your knowledge base or your lack of knowledge. If you have a lack of knowledge around all of these things, if you can't do any of these basic 4 things, you're going to be in real trouble. And, you know, when I look at this little picture, this is my dog Doobie dressed up as a bee. You know, when you think about bees, when you think about a colony, a lot of people don't think it's a big deal when they lose a colony. They just buy another one next year. I want to impress upon you that when you lose a bee colony is essentially like a 20 to 25 pound animal when you think about it as the superorganism. When you lose a bee colony, it's just like losing this little dog here, this 20, 25 pound dog, Doobie. When he dies, if your colony dies, it's just like losing one of them. And so you have to take this seriously. You should not be one of those individuals who wants to buy bees every single year. You should buy bees once, one time. And then from that one-time purchase, because you took the time to educate yourself on the front end, from that one-time purchase, you should be able to sustainably increase your apiary size every single year, just off of splits. So this is really important stuff. And what we're going to give you is the foundation here. Now, not to harp on this a lot, but I do like to harp on the fact that education is so important, because we don't need more beekeepers. And I think I've said it before, we need more educated beekeepers. And there is research to back this up. And in fact, in 2017, this is a journal PLOS One, a scientific journal. There was a research study that came out in 2017 from a group out of Europe. And they looked at almost 6,000 apiaries over 17 countries over the course of 2 years. And what they were trying to identify was, is there a correlation between education of the beekeeper and colony survival, hive health? Is there a correlation to that? So what they wanted to find out was, you know, do hobbyist beekeepers have the same success as professional beekeepers? Are they being plagued by the same problems? Maybe, you know, maybe the overwintering loss is the same for hobbyist beekeepers as it is for professional beekeepers. And that would say a lot. But the reality of that is no. There is no correlation. In fact, there is a correlation. Well, I should say this. There is a correlation between the education level and colony loss. And what their findings, this is just a summary of their findings. Hobbyist beekeepers with small apiaries and little experience in beekeeping had double the winter mortality rate when compared to professional beekeepers. Furthermore, honeybees kept by professional beekeepers never showed signs of disease, unlike apiaries from hobbyist beekeepers that had symptoms of bacterial infection and heavy varroa infestation. Our data highlight beekeeper background and apiculture practices as major drivers of honeybee colony losses. The take-home message is education is the key to success. It has nothing, these crazy overwintering losses that we have in the United States, 40% annually, more of this has to do with the fact that we have a bunch of uneducated beekeepers in this country rather than the diseases and the pests present. Varroa mite, there's no doubt that varroa mite is a global epidemic. There's no doubt about that. But my entire beekeeping career has been during varroa mite. And I can tell you with 100% certainty that varroa mite is manageable. We're never going to see it go away, but it can be managed. And it can be managed successfully. So the fact that we have varroa or that we have cold winters in Michigan, these are not excuses for colony loss. In fact, good beekeepers don't have a problem with winters. Good beekeepers don't have a problem with pests. We might lose a couple colonies a year, but usually there's something else going on. You need to understand that this education that you're providing yourself right now is going to be the key to this success. And it's why I try to instill upon you all that I know this is a lot of material I'm throwing at you, but beekeeping is not something to be taken lightly. It's just like any other livestock. And the thing is you don't want to be burying a 25-pound animal every single year from each one of your hives. I tell you what, when I have a dead out, and this year, you know, out of 20, I think we overwintered 22 colonies, we had 3 dead outs. One of them, well, 2 dead outs during the winter, and then one that just, it's not going to make it. It's a small cluster. And so I've counted that out. But 3 colonies out of 22. That was with me not being at this home where this apiary is at for almost 3 months during the winter. So there are ways to get things to survive, and it's just education, and that's what we're trying to provide y'all with. All right, so we're going to jump into what to expect when you get into your hives. So we're going to start with the brood chamber, and that is the bottom area there. Now, this generally, the size of a brood chamber is generally about the depth of 2 deep boxes. You can think of it kind of like, I always kind of say it's like the space of 2 basketballs or 2 volleyballs stacked on top of one another. That is about the normal size of a brood chamber. Generally, the queen will not go much beyond that. It doesn't matter what size boxes you're in. So in this picture here, you see 2 deeps. I generally run mediums, 8-frame mediums, and generally this brood chamber occupies about 3 to 4 medium boxes, but it's always going to be about the same size. So when you think about things like, people talk to you about, you got to have a queen excluder on because otherwise she's just going to lay in the entire colony. That's not true. The truth of the matter is that she will lay over a certain area, and generally it's about 2 deeps worth of space, or like I said, 3 or 4 mediums. So that is where the brood chamber is. Now, in the brood chamber, we've got a lot going on. And so let's take a look inside and see what you can expect to find when you open a brood chamber, those 2 bottom boxes. So the first thing you're going to see is bees crawling all around these frames. Now, in a brood chamber, there is a specific orientation that frames are laid out in. And this is an orientation that the bees construct themselves. Generally, there is going to be brood, eggs, and larvae in about the middle 4 frames on a healthy colony. So brood frames, larva frames, the brood includes eggs, larvae, and pupae. So we call that the brood area. Now, this is important to note, really important to note, because a lot of beekeepers, when we start out, the first indication that we want to get, the first thing we want to do when we're doing a hive inspection, most of what you're going to gather as far as data is located in the brood chamber. Whether you're queen right, what your patterns look like as far as your brood patterns look like, whether the brood is healthy, indications of disease, are there pests present? The brood chamber is also where we do varroa mite sampling from. So you're doing most of your data collection in this area, in these 4 frames. Now this would be, again, 2 boxes deep. So you generally have about 8 frames of brood stacked one on top of another. So they're stacked in a column. That's important to note. And the reason is because, like I said, most of your data collection is going to take place there, but it is not where you start your inspections. It is not where you start digging into the colony. And it's important to note what else is around here. And the reason I say you don't start digging in here, this is not the first place you take frames out of, is because this is where all the stuff is that's important. This is where your queen is. This is where your brood is. If you have queens being replaced, this is where the queen cells are going to be. So there's a lot of stuff going on here. And the last thing you want to do is to disrupt any of that. So starting here by digging into the colony right in this middle portion of the colony, that's the wrong place to start. I'm going to show you where the right place to start is. The next thing that's on the outside of your brood frames, is going to be a food frame. Now that food frame is generally one side of it is pollen and one side of it is honey. That's for the most part. Sometimes you'll have honey mixed in with pollen as far as on the frame orientation. But generally, the pollen is facing in towards the brood, and the honey is facing out towards the outside of the hive. The reason for that is that by this time, you all should understand that pollen is the protein source utilized to feed brood. And so that pollen needs to be very close to the brood, so that a bee that's a nurse bee that's feeding young can turn around, grab pollen, and feed it direct and be able to prepare that so that it can be fed to the brood. It has to be close by. Think about this in terms of clustering. So when it's cold outside, the bees cluster. The bees will not wander off several frames over to find food. The food has to be near the cluster. And so that's why this pollen is generally towards the inside, towards the brood frames. Because if they do cluster down, the place they're going to cluster is right on the brood, because the priority is to keep the brood warm. In those situations, there has to be pollen very nearby so that they don't have to break cluster to continue feeding. Otherwise, they can starve in place. They literally will starve, frozen right on that brood, if they're not able to maintain, gather food from nearby and maintain that colony temperature. It has to be about 90 degrees to keep that brood from chilling. All right, so there you're going to have some food frames. Now, outside of that, you're going to have your honey frames. And these are going to be solid honey frames that are on the outside of the colony. Now, you can see this is an 8-frame colony right here. Generally, if you have a 10-frame colony, you're going to have 2 more honey frames on the outside of this. This is the general orientation of a brood box. Both brood boxes will be oriented like this. This is the classic honeybee formation in the brood chamber. Now, getting back to inspections, where do you want to pull a frame out when you start your inspection? The first place you're going to pull out one of these frames is going to be in the first or second frame position. So in this situation, it would be this outside frame here, this honey frame, and you can see my cursor there, or this pollen frame. But generally on an 8-frame hive, you're going to want to pull this last frame. Why? Because this pollen frame is still interfacing with a brood frame. The idea is that you want to give yourself some space. So you remove a frame that definitely doesn't have your queen on it, which would be a honey frame. You're going to set that aside, and then that gives you room to pull this second frame back, and you can take it out as well, or you can continue to leave it in the box, and you have space to pull these frames apart as you're moving through them and pull them out so that the frames are not rubbing against one another. What you can do in situations where if you were to just pull out a brood frame, you can do what we call roll the queen, where essentially when you're pulling out the frame, a lot of times, although they are perfectly matched up when they're in there together, a lot of times the wax is not exactly perfect. And you'll get these kind of rises and dips in the wax. And as you're pulling those frames out, as they're coming across one another, some of those frames, because they're being moved out of position, will drag on one another. And you can actually roll a queen and kill her during that process. You can also roll a queen on the edge of the frame. So if you're not picking up a frame perfectly, in line, and you're dragging it, cockeyed against one side of the hive body, that could also be a potential place to roll the queen. So when you're pulling frames out of the brood chamber, the big thing to keep in mind is that your queen is in there, and you don't want to harm her. And so not only do you want to, pull out the outer frames first and then work your way in, you also want to be conscious of where you have that frame sitting. So when you're doing an inspection, the big thing to keep in mind is you want that frame over your box. It needs to be over a box, because if your queen drops, if she falls off the frame and falls on the ground, she's not going to find her way back in. She is not going to find her way back in at all. Unless you find her and pick her up and put her back in the colony, that colony is now queenless. And so it's better to do an inspection over your hive so that if she drops, she falls back down into the hive and not on the ground. I have seen many, many times where people get super excited about passing around frames to their friends. No, I do it when we would normally have workshops in Heroes to Hives. I do it all the time. We send frames around. But everybody's always conscious of where the queen's at if you have a frame with a queen on it. So if you're doing inspections and you have friends over and they want to see a frame, make sure you know if that frame has a queen on it. Because if she just drop off, that's a queenless colony at that point, and you've got to rectify that. So just keep that in mind. The other thing I've seen a lot of people do is damage queens. So they fall on the ground and you end up stepping on them or something like that. That's a pretty heart-wrenching thing, to find a dead queen by your foot. So keep that in mind as well. But this is the general orientation. All right, so the frames that you're going to find in the brood chamber. So this is now those middle 4 frames. This is what they're going to generally look like. This is a healthy brood frame. Now, I'm showing you what a healthy frame looks like. I'm going to also show you what an unhealthy frame looks like here in just a moment. Then we're going to also, at the back end of the lecture, going to talk about symptoms, and I'm going to give you some key things to look at. Now, I'm not getting into actual diagnosis in this lecture. We're just talking about the basics. But what you're going to leave with is this understanding that I hope you know what a good frame looks like. And if something doesn't look right, like it doesn't look like this, there could be a potential that something's going on. And I'm going to give you a few key symptoms to look for just to know that something's not right. It's not diagnostic, but it's enough to where you can say, okay, this doesn't look right. Now I need to get somebody that has a little more expertise to help me. That could be questioned through our Heroes to Hives YouTube channel. That could be, and I encourage those types of questions, but that could also be, and I'm going to go through this when I do a video inspection about what things look like and you know what a healthy and unhealthy looks like. But generally what you want to be able to do is just tell if something's right or if something's not right. And if something's not right, that's where you can get a mentor, someone from your bee club. You can send me a text message or an email with a photo in it. If you do that, please keep in mind, I'm not encouraging you to send me a bunch of emails with pictures, but if you do have something interesting that you're having trouble with, you can do that, and I will try to answer those. But I do encourage you, if you can post a picture on the YouTube comments, I'm not sure if you can, but I like to have all those things in the YouTube comments because everybody else can see it then. So anyways, but what I'm getting at is that you can understand that what looks good and what doesn't by the end of this. So this is a basic brood frame right here. And there's a few things that I want you to keep in mind. This shape, this kind of arched shape that you see here, this is a very classic arch to a brood frame. Typically, the queen is going to occupy most of the center of the frames with brood. So this is where she lays her eggs. On the outside of this, and let's go to the next slide here to give you a little more of a close-up. So this is our brood here, capped brood. And then right beyond that, we have this band of orange and yellow that you see here. That is pollen. So immediately outside of the brood area on a frame is going to be pollen. Why? Well, I said it earlier. They've got to have that pollen right by so that they can feed the young. And if they have to cluster, that pollen needs to be immediately near the young so that they can feed them while still in cluster. When my old mentor used to call this the power band. He said, you know, the bigger your pollen band here, and this can get excessive, and I'm not going to get into that right now because we can get to a condition where we're pollen bound, where the entire frame is pollen. But in this situation, we've got a nice strong power band, this pollen band right here. And so that is an indication that you have pollen in the landscape. If you did not have pollen on this frame, that could be an indication that plants are not putting off pollen at that time of the year, and you may need to supplement. An example would be at the beginning of the season when there's not a lot of pollen in the landscape. You may have soft maples flowering, you may have willows flowering early in the season, but other than that, there's very little food in the landscape. And so in those situations, if you were to do an inspection and see that there was no pollen, you know that you have to feed pollen, right? Because pollen is one of the critical components of rearing brood. It is the critical component when it comes to protein content. So just beyond this pollen band, you see this kind of like, you know, you can see that this is something that's capped, but it's this real kind of raggedy cappings. That is honey. Now, the big difference between honey and capped brood, and I get all sorts of questions about, is this brood or is this honey? The way you can tell the difference is that bees spend a lot of time on their brood. A lot of time. And so when you look at the brood cells closely, what you can see is you can see each individual cell is individually capped. You can see a distinct cell with every single one of these caps. That is brood because they are capped individually based on when the egg was laid. You may have one row that was laid 10 hours before the next row. They're going to cap that accordingly to age. So these are laid individually, which is why there are distinct lines around each individual cell. When you look at the honey up here in the corner, you cannot distinguish between any of those cells at all. The reason for that is because bees, when they are capping honey, they cap in layers. So they just are capping an entire section at once, rather than each individual cell, which is why you have such a distinct difference in the profiles of the tops of those cappings. Very, very distinct. So you should easily be able to tell which one is which right now, just by looking at it. And when you get into your hives, that's going to be the distinct difference. But this is the orientation, classic orientation of a brood frame. Now, just to kind of ram this home, we want to take a little bit of a closer look here. In this picture, you can see the two types of brood that you're going to have: sealed brood, which is the capped brood, and open brood. So you may look at this frame and you say, oh, well, there's this big open area right here where there's nothing in there. But if you look closely, you can see in each one of these cells, even the small ones up here, there is this white substance in there. That white substance is royal jelly, or food that's being fed to these are very young bees. So this is potentially royal jelly right here, but it's bee food. It's the food that's being fed to the bees. It's essentially a protein-rich jelly that is being created by the bees and fed to the young to help them be reared. Now, when we look at all of this, going back to here, you can see that some of these are big white cells right here. There's a lot of big white stuff in here. Those are instars, and I'm going to get to what an instar is here in a little bit. Those are instars, which are the young larvae. Those are just more advanced than the others. So these are a little bit older that are in this section that are the big white cells, as opposed to the ones out here on the edge, which if you train your eye, you can see there's eggs in this outer area. And I'm going to show you another picture later on of what eggs look like. But essentially, this is all stages of brood right here. You have everything from the egg stage on through the second instar, third instar, all the way to sealed brood. In addition, you see the pollen band here as well, and the honey layer. And here's a really good, nice close-up where you can see that how that honey is laid, the cappings to that honey is laid down in comparison to our sealed brood, where you can see those distinct cappings for each individual cell. All right, so honey and bee bread. So I'm going to start with nectar. So nectar is collected from flowers, and it's converted by honeybees into honey. So nectar comes in, and it's a complex sugar, sucrose. It's broken down with enzymes in the salivary glands to produce fructose and glucose, which are very simple sugars. Honey is a combination of fructose and glucose. Honey that is brought in, or nectar that is brought in and converted by enzymes, by breaking it down, is then placed into cells and allowed to dry. Now, when they place honey, we'll call it, it's nectar at this point still because it has a high water content. When they place this nectary, let's call it a watery honey, in the cells, the way that they do this is they actually wipe it on the outer edges of the cell. They don't just deposit it in the bottom. They wipe it on the outer edges. And why they do that is to give it more surface area to allow it to dry faster. What they're trying to get to is once this nectar substance hits an 18% water content as honey, then it is true honey, has an 18% water content. That essentially, at that point, it is then capped, and essentially at that point of 18% water content or less, that honey can stay in that state forever. Is at 18% water content. It will not ferment. Honey that has a high water content, meaning that it has not been capped, will ferment over time. You as a beekeeper may have experienced this at one point or another, or even as a honey consumer, where you purchase honey from a beekeeper, you bring it home, and maybe right away or over time, it starts to smell like alcohol. What you're seeing there is a fermentation process where sugar is converting into alcohol over time. Now, as a consumer, we do not want fermented honey generally. We want honey to be honey. If you harvest honey that is uncapped, you are harvesting high water content honey, which will ferment over time. Or if you leave honey setting out, honey absorbs water in the environment, it will add water content to it and will ferment over time. So it's why processing and handling is so important when it comes to honey. And we have a whole lecture on processing and handling related to honey matters that we'll come to at the end of the season. But that is honey. It's essentially nectar that's coming in, that's converted into simple sugars, and then dried down to an 18% or lower water content. Now, pollen. Pollen is a little bit different. So pollen's also collected from flowers, but it's also modified by bees because pollen has a really high moisture content. And if it's just stored as it is collected from flowers, it's going to mold pretty darn quickly. It's going to mold. So when bees bring in pollen, they combine it with digestive fluids that are really, so it's digestive fluids that are really rich in this lactic acid bacteria. So it's this bacteria that basically metabolizes the sugars in the pollen and it produces lactic acid. So the bacteria feeds on it, it produces lactic acid. And what that does is that lactic acid content actually drops the pollen pH to about 4.6 or so. Now, at 4.6, that's generally the kind of the threshold for things to be able to mitigate bacterial or microbial growth, we should say. So nothing grows on it. It's not going to mold. It's not going to, really have any of those I shouldn't say fermentation because it is a fermented food. It's not going to have any microbial growth on it at that pH. So what they do at this point, after they've added the digestive enzymes and the bacteria that basically converts these sugars into lactic acid, after that happens, they then seal this with a drop of honey. And so in this state, this is now a fermented food. And we call this fermented food bee bread. That is what bee bread is. It is not just pollen. It is a fermented food. Now you can see in this picture here, the top picture, you can see that you get these wild colors and you can see this has been sealed with honey. You can see the shiny surface on it. And pollen's not going to be all the way to the top. They generally fill it, about 3/4 of a way, the cell, and then they cap it with that honey. And so that shiny surface, that is honey sealing it. When you look at this from the side in this second picture, you can see that this is literally just pollen grains that are kind of been macerated and packed on one another. Very colorful. But that's bee bread right there. And this is important because this is the protein source that is fed to our young. So a very important thing. The thing to keep in mind about pollen, and I know we use pollen substitute, this time of year, even my bees are feeding on pollen patties right now. Pollen patties are important because we have times of the year, particularly in places like Michigan, where we don't have pollen available, but bees are moving on. And we need to be able to feed them supplemented pollen so that when there's, bad weather, and, the UP is a prime example. If I have 2 weeks of bad weather, they still got to be able to rear their brood. They don't stop because of weather. They've got to be able to continue what they've started. And so you have to supplement them. But the thing to keep in mind is pollen that is fed as a protein patty is not stored pollen. So the stored pollen is the stuff that's coming from the environment. And it's really important that you have stored pollen. Like we talked about when we were on this brood frame here, there's a lot of stored pollen on there. That's the indication that they can feed a lot of young. If there was little pollen on here or no pollen on here, they're not going to be able to feed anybody. And so that's when you have to supplement. The one last thing I want to leave you with pollen before we jump into healthy brood frames is this. There's been a lot of research that's been done on pollen. Pollen is not something that keeps. So even though this is a fermented food, it does have a shelf life. And generally, pollen that is collected within a single season and utilized during that season has very high quality properties to it. But as time goes on, that pollen degrades. So for example, if you have pollen frames from a dead out from last year, although that pollen may still be, looks decent, it does not have nearly the nutritional value as fresh pollen. So just keep that in mind. I mean, you can feed pollen substitute in those situations as well. But I'm trying to instill upon you that pollen does not have an indefinite shelf life, a lot like honey does in some situations. But it is something that is very important for bee nutrition. And you should as you're going through your inspections, you need to note, just, mental note, oh, this has a lot of pollen in it. Great. If there's no pollen, something's going on. So simple things like that to help you understand what looks right and what doesn't. All right, this frame here, this is a healthy frame right here. And what I want you to look at here is you can see that around so we've got a nice healthy brood chamber right here. We've got a band of pollen, a nice band of pollen around. And if you really look closely, it's kind of hard to see in this picture. When I look at my big screen, I can see here that, you know, there's shiny things in these cells on the outsides. That's honey. There's also shiny things on these cells on the inside. You can see here. That might be honey too. If that is the case, that's backfilling. But a lot of times with these types of pictures, it's hard to tell because you can see over here on the edge, we've got white. Brood is always going to be healthy white. If it's not healthy white, something's wrong with it. If it's off color is what I'm saying. But so you've got a lot going on this frame. But essentially, this is what a healthy frame looks like. You've got a lot of brood on there. Again, she's not going to miss, or, you know, I may not have mentioned this earlier, but she's not going to miss any cells, the queen. She's very good at what she does. If she's missing cells, there's something going on, whether it's, you know, backfilling from workers that are trying to find space to store honey, or it's that you're getting spotty brood because you have a lot of larvae that are dying over the course of being reared. So that's a healthy frame. This is an unhealthy frame right here. So this is a frame that when you look at it from here, you can see that it's very spotty. There is some pollen up in here. You can see that there's pollen stored here. I can also see that there's a little pollen stored here at the bottom as well. But for the most part, there's a lot going on here. And one of the things that I'm going to show you here at the end of this lecture is some symptoms. And we're going to come back to this frame. And I'm going to give you a snapshot of what's going on in the middle here. But you can see that there's stuff in these cells if you look really closely. I'm going to show you that this stuff that's in these cells is not healthy brood. And we're going to look at an example of healthy brood and an example of unhealthy brood. But when you see this kind of pattern, this open pattern like this, where it's not, you know, go back to the last page where it's solid brood. When you see this open pattern, there can be a couple things happening in a situation like this. One, you could have just had a whole bunch of brood emerge and the queen's probably going to come back through here and fill it. Or two, if you look at it and the brood that's in there is not healthy, that's a situation where you may be dealing with a bacterial infection or perhaps parasitic mite syndrome or something like that. But again, we're going to come back to that when we talk about symptoms. I just wanted you to start thinking about what looks right and what doesn't. Okay, so we're going to continue on with what's in the hive by going through what bees you can expect to find in your beehive. So this is the castes and sexes of your bees. Now, there's going to be bees crawling all over the place when you're doing your hive inspection. All over the place. In a mature hive that's mid-season, 60,000 bees, and there's one queen in those 60,000 bees. There's also drones and a whole bunch of workers. But you are going to have to be able to identify the difference between all of those bees as far as their sex and what their responsibilities are. So let's start with the worker. You can see my chart up here in the top right corner where we give you the size of the individual workers and drones and queens. So first thing you should note is there is a big difference between sizes between these three bees. Now, when we talk about the workers are all females. And they are the most numerous, and they are the smallest in the colony. So there are the most of them in the colony. And in fact, you know, primarily the entire colony is made up of workers. And at some time or another, there's some, you know, during the season, depending on the time of the season, there's also drones present. But when it comes to workers, you need to be able to look at a bee and be able to say, okay, without a doubt, this is a worker. How do you do that? There's some key characteristics on these bodies that can help you determine that. The first one, and the easiest one, look them in the eyes. And I don't mean that to be funny. I mean that, well, I kind of mean it to be funny, but I mean it more because you need to look them in the eyes. Worker bees have distinctly smaller eyes than drone bees. Now keep in mind that the queen is also a female, so she has the same eyes as the worker. But when it comes to a difference between drones and workers, workers have eyes that occupy the sides of their heads. And so you can distinctly see on this picture on the left that the eye only occupies the side of the head. In addition to that, she also has a distinctly tapered abdomen. The second picture that we see here, that she's foraging on clover, you can see that the abdomen tapers down to a point. Why do you think that is? Probably because there's a stinger in there. So it tapers to a point where the stinger is. That is the overall body morphology of the worker that is important to understand to distinguish between the two. So when somebody looks at a colony, yourself included, and you're looking to identify, is that a drone? Is that a worker? You should not be staring at the whole bee. I mean, you can, but you should be looking them right in the eyes first. That should be your first thing you go to, is look them in the eyes. And here's why. Because this is a drone. What you see on the left is a worker bee, again, with the eyes distinctly on the outside of the face, of the head. The male drone, this one right here, you can see the eyes occupy the entire head. The entire head. Why would that be? I'll give you a second to think about it. Why would that be? We've talked about drones in previous lectures. We've talked about mating and how mating happens in flight. Well, wouldn't it be useful for a drone if he could see 360 degrees when there could be a queen flying by him that he needs to go and mate with? That's exactly why their eyes are so big, is because they are literally mating in flight. They are finding their mates in flight, and they've got to be able to see them. Evolutionarily, this makes sense to have big, huge eyes when you're a male honeybee. It does not make sense to have big, huge eyes when you are a female worker bee. So that's why we have these big eye differences. When you look at these two in the face, like I said, look them in the eyes. When you look at these two, they are 100% different looking. There should be no mistaking one for the other. And so when you get into your colony, or when you get into your friend's colony, or whenever you interact with bees, whether you see them in the landscape or not, you can without a doubt tell your friends and family when you see a honeybee, fly by on a summer day and land on you. You can definitely tell them if it's a male bee or a female bee just based on the eyes. Now, in addition to the eyes, the side picture here, you can also see that the abdomen is truncated, meaning, well, it's blunted, I should say. It's definitely long. I mean, just kind of, it's actually quite robust, the abdomen. But you can see that it's blunted at the end, and that it's also furry at the end. Why could it be blunted at the end? It doesn't need to be pointed like the stinger. Well, the number one reason why is because they don't have a stinger. They have what's called an ediogus. And an ediogus is essentially a penis. So instead of a stinger, they have an ediogus. Now, what does that mean for you? Well, number one, if you see a drone flying around, you can happily pick that drone up. You can let children handle it, and they're not going to get stung. One of the things that we used to do when we would do direct marketing, my wife and I, is we would oftentimes bring it's escaping me right now, an observation hive. We would bring an observation hive to it when we went and did farmers markets. And a lot of times I'd take out a few drones. Now keep in mind, drones don't live very long by themselves because they do nothing. They essentially don't feed themselves. They are fed. They really only have one purpose, and that's mating with the queen. So they don't really do much, and without a colony, they will only live for a few hours. But what you can do is we used to let people feed the bees. And so, I'd give a little drop of honey on a kid's finger and put a drone on there, and the drone would eat the honey, and the kid would go crazy because they were holding a bee. And it was like the most special thing in the world. And mom and dad would usually buy a bottle of honey after having that experience with a honeybee. So it's something to consider, not that I'm trying to tell you to do that as part of your marketing techniques, but it's just to illustrate that drones are safe to handle. We talked about, I think in one of the Q&A sessions, about marking queens, and I'm going to get to that in this next slide. If you want to learn how to mark queens, mark your drones. Don't practice on your queen. Practice on your drones, because you're not going to get stung, and you're not going to damage the queen if you're practicing on drones. So that's a little bit about the drone and how to tell the difference. You all should be able to easily tell the difference between the two now, between workers and drones. Queens. So this is where the real problem happens, is people trying to identify queens. The thing about queens is she is a female, so she looks a lot like the workers. And you can see her, she's the one that's tagged with this green dot right here in this picture. You can see all these bees, worker bees facing her. Those are her attendants, because she is taken care of as well. One thing to note about the queen is that she is the longest bee in the colony. And generally her abdomen, when she's fully gravid, meaning that she's laying eggs and, you know, full of sperm, when she is fully gravid and really going to town, that abdomen can oftentimes be almost as long as a worker. Very long. So the big distinguishing factor is the length of the abdomen, being able to determine that. That can sometimes be hard to do. It can really be hard to do for beginners. So a lot of beginners like to have their queens marked. Now, a lot of professionals also like to have their queens marked. So, you know, for myself, for example, we mark all of our queens, not because I have a tough time finding the queen. I can generally find a queen very quickly. The reason we mark them is because this is how we keep track of how old the queen is. And so you see right here in the bottom right corner, this is the, and I've alluded to this before, but we have not gone over it, I don't think, in any of the previous lectures. Excuse me. But this is the color coding that we use internationally to mark queens so that we know how old the queen is. So when you have a queen that is white, that means that the year that she was born either ended in a 1 or a 6. And that goes on to different colors. So the coding combination is white, yellow, red, green, and then blue. So it's 2020. What color are we? You're blue. The same color we were in 2015. This rotates every single year. So last year we were green queens. The year before that we were red queens. The year before that we were yellow queens. This is a great way when you have many colonies to be able to determine how old your queens are very quickly. And I'm not going to say that it doesn't make it easier to find her as well. I mean, it's obvious if you have a red dot staring at you on a frame, that makes it a lot easier to find her much quicker. But it is also, you are also able to find her unmarked. So don't think that you have to have your queens marked. A lot of queen rearers, you know, people you buy your queens from, or people you buy nukes and packages from, will give you the option of marking queens or not marking queens. But you can do it yourself also. Generally, I think the going rate's about $5 to mark a queen. So if you want to save yourself $5, you should learn how to do this. You should learn how to do it anyways, because it gives you a good idea of how gentle you have to handle the queen when you're doing this process. Normally, you know, I don't, and I said it in the last slide, I don't recommend you trying this on your queens if you've never done this before. I would encourage you to try doing wing catches, which is, I think we've talked about it in a previous, I think it was a Q&A session, where you essentially follow the bee along and you catch the two wings when they bring them together on their back. And then you were able to manipulate the bee from there. And where you essentially get to when you're marking a queen is you want to have her thorax held between your index finger and your thumb. And that's the hardest part of her body. So you can hold pretty firmly on the thorax, and then you take your pen and you just dot her right on the back, just like this dot is right on her thorax. You always want to dot the thorax. You don't want to get paint on the wings or the eyes or any of that kind of stuff. Another thing also with marking, and I'm going to stop talking about marking because it's something that, you know, if you're a beginner, you're not likely doing. But one thing to keep in mind is you also have to let the paint dry. I've known a lot of folks that will mark her and put her back in the hive immediately. If that's the case, usually they just lick off the paint and you don't have a marked queen at that point, which can be confusing for some folks that you're sure you marked her, but she's not marked now. And now you've got an unmarked queen. You think, oh, what happened to my queen? Did she, you know, was there a super seizure or something like that? But generally you got to let this dry. So you got to let her sit out for maybe not 10 or 15 seconds. Sometimes I'll blow on her to get it to dry faster. But that is the queen. She is also the center of the entire colony, right? She is the one who is laying all the eggs. It's very important to note that generally we replace queens about every year. And that's for productivity, because once you get a good season out of a queen, she's never going to be that productive again in general terms. Not to say that I haven't had queens that have been 2. Sometimes I may have in my career had a 3-year-old queen at one point or another. She would have to be super productive if that were the case. But most of the time you replace them annually. So don't get too hung up on your queen. I know a lot of you get really concerned when it comes to genetics. as beginning beekeepers, don't get hung up on what genetic line your queens are. You just want healthy queens. It's more important to have healthy beehives than it is to get hung up on the nuances of queens. But you can buy fancy designer queens. There's a lot of good genetics out there. Some of the genetics that I've used in the past, Italians, Carnies, are kind of your run-of-the-mill bees that are just available everywhere and pretty gentle. More recently, or I guess in the last probably 7 or 8 years, we've gotten into some varroa-sensitive hygienic queens. So Purdue ankle biters we've run before. This year we're running pole line queens that are a varroa-sensitive hygienic queen that was bred out of a USDA program down south. We've used Saskatraz bees, which were bred for winter hardiness up in Saskatchewan. So I go through a lot of different queens. I like to try all these different things out, but I've been doing this a long time, and so you can see the nuanced differences between them. Most people are not going to notice those things early on. So your management of your colony is much more important than the queen that you have at this time. So don't get hung up on the genetics until you get a little more of a better understanding. And what I mean by nuanced differences, so I'll give you an example. Saskatraz queens. Saskatraz queens generally start laying very early in the season. And that colonies that, and this is from personal experience, colonies that have Saskatraz queens in them will swarm earlier than any other colonies because they put on brood so quick in the spring. So it's kind of like as soon as they get the green light to start laying brood, it's off to the races. Whereas with other colonies, they generally start a lot slower. So other lines of bees, like your Italians, will start slower in the season. And so they'll ramp up and you'll have a big increase at some point. It's just delayed from Saskatraz bees, from my experience. The other thing I would say about Saskatraz queens and bees in general is that normally you have a pretty good die-off in the winter. You have a lot of winter bees that will die over the winter. Saskatraz bees, they do not seem to have winter die-offs. Like when I clean my bottom boards out of my Saskatraz colonies, they're generally pretty clean, whereas in most of my Italian or Carney colonies, there's a layer of dead bees on the bottom board. So just something to note. But these are nuanced differences a lot of people just aren't going to see in the beginning. So don't get hung up on it. All right, so if you can't find your queen, you can always tell if you're queen right by finding eggs. And this is what an egg looks like. You as a beekeeper, you have to know what this looks like, because you are not going to want to spend an hour trying to find a queen in a colony. Your job when you're doing an inspection is to get in and get out. Get the data you need, have the least amount of disturbance possible, and then get out of that colony because you don't want to rile them up. So by seeing eggs, if you can see eggs in a colony, and they're always going to be 1 egg per cell, perfectly positioned. We talked about that in another lecture where we talked about laying workers, but you're going to see them perfectly positioned at the bottom. That's very important to note. You see these eggs, you know your queen right. You can say that your queen is there and she's laying, because we know that eggs hatch after 3 days. So if you see eggs, you know a queen has been there in the last 3 days. And that's a good indication that you're queen right. Again, you don't have to see the queen to tell that your colony is what we call queen right. You just have to see eggs present. All right, so that's what an egg looks like. You should all that have bee colonies be able to tell what this is. If you cannot see eggs because of eyesight or whatever, get a flashlight. Point it down into the cells and you'll see an egg. Another trick, if you have trouble seeing eggs, buy black foundation for your brood frames. That will help this white egg pop. But you have to be able to see this. The other trick, and because we aren't having, we're not going to have an experience together this year doing this, the trick to seeing eggs is that you have the sun to your back coming over your shoulder and the frame in front of you, and that sun is shooting right down into the cell. You may have to move it a little bit to line it up right, but when you do that, you should be able to see eggs very clearly on a sunny day. So there's your eggs. What else are you going to see in this brood area? And we're still in the brood chamber, right? Still in the brood chamber. You're going to see eggs and larvae. Now, this is the timeline of a young honeybee. This is the egg stage through the larval stage. So what you can see is the egg, which we talked about. Here's a cell right here with an egg. The egg that we talked about, that is the first stage of the bee's life. It goes through the egg stage for 3 days. Then it hatches into what we call a larva. Larvae have, with honeybees, have 5 different stages, and we call the 5 stages, those 5 larval stages, instar stages. This is the same terminology we use with any kind of, with any insect. That is a insect that undergoes complete metamorphosis. What do I mean by complete metamorphosis? Well, it's the same thing as a caterpillar. It goes from an egg, it turns into a larva, and then it goes through a pupal stage, and then turns into something, the adult stage, that is completely different looking than the larval stage. Here, you have what looks like a grub. It's a little kind of worm-like larva. That larva is going to turn into a honeybee at some point. But in this stage, in the immature stage, we call, you're going to have this larval stage. So when we look down into these cells, you can see all the different stages represented here. So right here, this cell right here, that is a first instar. And you can even look at the picture. You see there's a little tiny piece of what looks like a little tiny piece of rice down in that kind of bed of jelly right there. Then we have the second instar. The second instar is distinct in that it is kind of like a crescent-shaped moon, right? So it looks like a crescent right there. So if we go over here to our cells, let's see if we can find one. So you've got one right here. So there's our first instar. You've got one right here that looks like it's just about turning into that second instar. It's got that crescent kind of going on a little bit more. The next instar is the third instar. And that's this one right here, where it completes basically a C shape. And then you have the fourth and the fifth instar where it basically just fills out. And by the time it's so here we have like right here, maybe like right here, we'd have a third instar, fourth instar, fifth instar, you can see that it's just filling that entire cell. These are the 5 larval stages of your bees and the egg stage. That is the first half of its life. What happens from here is then the brood, once it hits that 5th instar and starts to pupate, it goes through I'm tongue-tied, pre-pupal stage. That's when things get capped. So once it reaches the end of that larval stage, those cells are capped and the larva starts pupation. Pupation is where it changes. It's a quiescent state. So it's basically like a quiet state where they go from being a larva into being an adult bee. And they're capped during that process. Nothing is being fed to them. They are just going through metamorphosis at that point. So the attendants, the nurse bees, are not continually feeding them at this point. They're just going through metamorphosis. Now, there's 2 types of capped brood. I showed you in the pictures before what capped brood looked like on a frame. These are the two types of brood that you're going to see. And there's going to be one more, and I'm going to show you that here in just a moment. That's the queen cells. The 2 types of capped brood that you have are worker brood and drone brood. Now, when you look at them side by side, you can tell they're different. The drone brood is always a bigger cell, and it has kind of this dome cap to it. Why? Well, it's because the drone's a bigger bee. We talked about that just here a moment ago in the sex and caste section. It's a bigger bee. It needs more room. And so you kind of have this like cocoa puff top to it. That is drone brood. The worker brood is always kind of a level cap to it. Now, in both situations, you can see that this should not be mistaken with honey, right? Because the cells are placed individually. They're capped individually, so you can see that outline of each individual cell, as opposed to honey, which again just kind of looks like a wavy mess over the top of it. These are your 2 capped brood. So from here, the next stage that you will see your bees when they come out of this is as an adult. They will come out as an adult bee. They'll be very furry. They're very cute when they're born. Just like all young things generally are pretty cute when they're born, but they're furry and cute. And that's your adult bee as they start to emerge. The other thing you may see in your hive are queen cells. This is a queen cell. So a queen cell, this is a capped queen cell, I should say. A queen cell is where, well, number one, where a queen is being reared, but number two, has a very distinct look to it, right? It looks kind of like a peanut almost. And queen cells, so you see how this is oriented. This is a cell that's coming off of the frame, kind of like, say this, my hand here is the frame. It's coming off of the frame and hanging down towards the ground. Why is that? The reason that is that queens are known as, well, they're called, well, trying to get these terms out, they're gravitropic, meaning that they are sensitive to gravity. So, as an example, they're always facing down, always facing down. If you were to flip this over and to hold it so that the cell was facing up, you could kill the queen just by doing that. You would never store a queen cell up because she is gravitropic. Gravitropic, meaning that she is sensitive to gravity. You can kill a queen that way. It's why handling frames with queen cells on them is so important. Number one, they're very delicate. They hang out from the frame. So like I had mentioned before, say, for example, you just decided I'm going to go for the brood chamber when I come in and do my first inspection. I'm going to go right for that center frame, and I pull it out without taking out those outer frames out first and giving myself some space. This is why you don't do that. Because if you have a queen cell that's hanging off the front of your frame, I will guarantee you will kill that queen or damage that cell in the process of taking out that middle frame. It's why taking outside frames out first are so important. It's because you're trying to maintain the integrity of what's going on in that brood chamber without causing a bunch of ruckus or a bunch of damage. But these queen cells, they hang out pretty far, and you can easily damage a queen cell by not taking out those frames from the outside out first. Now, when we have queen cells, it means all sorts of different things. And this is not a lecture that's going to be in-depth on queen cells. We are going to go through the three types. But generally, there's two different situations that happen, and people freak out about the first one. The first one is the queen cup, which you see here on the left. Queen cups are normal. Queen cups will always be in your bee colony. They're just there in case. They need to rear a queen. There's a cell created for it. The queen cup means nothing because it has nothing in it. So a queen cup is a queen cell like this that has nothing in it, meaning there's no egg in it. There's no larva in it. It's just a cell, nothing to get worried about. Now, if there is something in it, meaning an egg, which you should be able to look for, right? With either a flashlight, maybe you get them out of magnifying glass, however it works for you. Get your bifocals out. I mean, if you gotta have it close up, you need to have those on. But if you see an egg in here, it is no longer a queen cup. It is called a queen cell at that point because they are actively raising a queen in that cell. So that's the difference between a queen cup and a queen cell. A queen cup is nothing to get excited about. A queen cell is something maybe to get excited about. Maybe you're raising a queen and now you've got a queen that's being raised, or maybe you have a queen that failed and they're raising another queen. That is something to note, is when those queen cups have something in them. Don't get freaked out, because I get calls all the time. Adam, I've got these queen cells all over the place. And I say, do they have anything in them? And most of the time people say, no, I don't know. But if they go back and they say, no, they don't have anything in them, I say, well, there's nothing to worry about. It's just a queen cup. So queen cell, that's what they look like. We showed you that on the last slide. So this could mean two different things, right? Okay, so we have a queen cup that has an egg in it. That's now a queen cell. When that queen cell is open, meaning that it has not been capped yet, like the queen cell on the right, we call that an open cell. It means that the queen is still actively being raised. Just like the worker, she goes through instar stages. Those 5 instar stages are important. She's being reared through those instar stages just like the others, and being fed just like the others. So it's open. They have access to her, to feed her. Now, this is the other situation that I get. Now, this will be capped, and then she will be reared, and you will have a queen that will hatch. And we've talked about the life cycle of the queens in past lectures. But generally, what I wanted to point out in this lecture was we have two situations that we oftentimes see open cells. One of them is when it's a truly open cell, meaning that there's a queen in there and that she's being raised. And the other one is a hatched cell. So oftentimes I get calls where people say, Adam, I have a queen cell, but there's nothing in it. And it's a really big cell. And I say, well, can you tell me what the outer edge of the cell looks like? So you can look at these two cells here, and you can see that the outer edge of an open cell that's active is quite smooth. Whereas a hatched cell, meaning a queen has hatched out of this, which you will find in your hives, especially if you have a hive that's swarmed, you'll come back and you'll find these cells all over the place. And you'll say, oh my gosh, there's all these queen cells, but there's nothing in them. The way you can tell that this is hatched is you can see that this edge around that cell is ragged. That's because there was an insect chewing its way out of that cell. So it's ragged because it's been chewed on. You may or may not have the cap attached here. I like this photo because it's got the cap attached still. But that may or may not be there. But the fact that this is ragged around the edge tells you that this is an old cell, whereas this is a new cell. So those are a couple situations that you'll see with queen cells in your colony. Okay, so we're gonna kind of wrap up here going through some queen cell stuff, and then we're gonna talk about symptoms that you should be looking for when you're doing inspections, just to tell if something's right or not. Again, this is not a comprehensive inspection lecture. We're going to do that as a video when I'm going through my colonies here. This will be at the end of May. You'll get this, I don't want to call it a lecture, but our workshop video. All right, so queen cells. There's three different situations in which you have queen cells. Now, queens are reared in all sorts of situations, and there's reasons for each one of them. And we're going to kind of just go through an overview of queen cells and what are some of the things that you would see that would lead you to believe that one is one type over another. Okay, so we're going to start with supersedure cells. So supersedure cells they're essentially the colony's way of replacing an existing queen, really due to something like, I don't know, sickness. She's old, she's not being productive anymore, maybe she got hurt. That's when they're going to replace her with a supersedure. So meaning that she's still in the colony, but something's wrong with her. Now, this typically, when this happens, what we typically see is we see a small number of cells. So I use the one to three cells. It could be a few more. Well, it's not going to be less than one, but it could be a few more. But generally, it's not a ton of cells. It's just a few queen cells throughout the colony. And they're generally built anywhere on the frame, because what's happening is they're taking one of those queen cups. The queen has laid an egg in there, and they're rearing a cell from that queen cup. That is your typically supersedure procedure. Other than that, there's really not a lot more to it. The big thing is that there seems to be a few less or less of them, right? It's just not, there's not a ton of queen cells everywhere. That's a typical supersedure. Swarm cells, very different. So swarm cells are a method of colony reproduction. And this is going to result in half of the colony leaving with the old queen and the other half staying with the new queen. So this is natural reproduction. And these cells look similar to supersedure cells. There's just more of them. And like you can see on this picture right here, there's all these queen cells all over the bottom of this frame. When I think of swarm cells, I typically think of cells that are lined up on the bottoms of the frames. And the reason is that they're just trying to find space to put these cells anywhere. What I do when I'm generally in swarm season, and one thing, this is a neat thing I'm going to share with y'all. Megan Milbrath and I were talking with Anna Heck and Dan Wines, the rest of our apiculture team, last week. And she made a really interesting comment that she'd look back at some environmental data that we collect at Michigan State University through our EnviroWeather program. And that apple blossom tends to be right around swarm season. Right around, always when you're right at apple blossom, it tends to be right around swarm season. So it's something to think about. But anyways, I digress. So really what you're looking for when you're looking for swarm cells is it's very similar to a supersedure situation. You still have a queen in the colony, so it's not like it's an emergency situation where you have no queen. But they're going to be kind of all over the place. What I always think of when it comes to swarm cells is there's just a lot more of them than supersedure cells. And when I'm checking for swarms to see if my colonies are getting swarmy, they're filling. So you're looking for things like backfilling and signs like that. But a lot of times I'll just pop open the colony in the brood box and I'll just look at the bottoms of the frames. If I see a bunch of cells being built out on the bottoms of frames, I typically know that I'm getting ready for a swarm. And right at that point, I'm actually well into the swarm impulse. It's going to be hard to mitigate that once you're got to that point where you have that many queens being raised. But generally, you're just going to see a bunch of queen cells when it comes to swarm cells, to swarming. Emergency cells. Okay, so an emergency cell is a different situation than a supersedure or a swarm procedure. This means that the colony is replacing a lost queen. There is no queen in the colony. And so what this means is that the queen cannot lay eggs into swarm cells at this point, or into queen cups at this point. They have to rear a queen from an existing egg. Now, you can change from a worker to queen while you're in the egg stage, up to that first day of larval stage. But once you get past that, it's too late. So what they do in this situation is they actually build off of an existing worker cell. So the cell was built for a worker. There was an egg laid in there. The queen died somewhere right after she laid that egg, or maybe she died because you did an inspection and crushed her, or maybe she fell out of the colony, or maybe she just died, for one reason or another. But essentially what they're doing is that they're not rearing a queen like they would from a queen cup. They are rearing it from an existing cell. And what they're doing is they're just altering the diet to a diet of royal jelly so that they can create a queen from that worker cell. This is a nice kind of way to be able to, recover from a queen being lost. This is, and these are the three typical situations you're going to find yourself in. The swarm situation is very common with people who are not managing space very well. So you got to super, and you got to super often when you're in the height of the season when a lot of honey's coming in. In addition, you always have to keep in mind that queens get hurt sometimes. Maybe you didn't kill her when you were doing an inspection, but you pinched her pretty good, and she's not going to be able to survive for much longer. That would be a supersedure situation. So all these different situations for different situations the bees find themselves in. All right, so we're going to go into symptoms now. And again, this is not a lecture meant to give you a comprehensive understanding of how to diagnose everything in a colony. What I'm trying to get you to the point to do is to understand what healthy looks like and what unhealthy looks like. And when you see unhealthy, that means that you have to hit the books, right? That means that you should be pulling out diagnostic guides for determining honeybee diseases, which are available. Purdue actually makes a pretty good one. And so you need to have some sort of a diagnostic, or you refer to a reputable beekeeping source like your books. I think mine are stacked right behind me. You hit the books and you try and determine what you're seeing, but be smart about it, right? You don't need to spend a lot of time on something that's healthy. And so that's what I want to get you to today, is I just want you to understand what healthy looks like and what unhealthy looks like. And there's a very typical unhealthy, and then what is always a typical healthy. So back to our frame. This is the frame I showed you early on, and I said this is not a healthy frame. Now, I had told you that this could be a situation where there was a bunch of bees that just hatched, but even from this picture, I can tell this is not a healthy frame. Why? Because there's some stuff going on right in here, and we're going to get a close-up here in the next slide. So this is that same picture on the left that I just had in front of you. And now we're much, much closer. Now, granted, my camera is not as good as I'd hoped it'd be when it got in there, but you don't have to see a lot to be able to tell this is unhealthy. On the right is the healthy frame. This is from our picture earlier where we showed all the larval stages. And you can see very distinctly, and I didn't mention this in that first slide that we talked about the larval stages, but you can see that always, 100% of the time, a healthy colony has larvae that are white in color, bright white in color, and rest in a C shape on the bottom of the cell. 100% of the time. If you do not see this on the right, something is wrong. Something is wrong, plain and simple. If your larvae are not in a C shape seated at the bottom of the cell, you can see that they are not tilted. They're not laying on the bottom of the cell, or I'm sorry, they're not laying on one of the sides of the cell. They're laying smack in the bottom of the cell. Any deviation from that, something is wrong. Okay, let's go to our unhealthy frame right here. You see white stuff in the bottom of these cells that look like they're kind of trying to make a C shape. The one where my cursor's on right now has a distinct brown color to it. The one next to it has a distinct yellow color to it. The one above it, right here, there's just a mass of white laying on the side of the cell. This colony right here is sick. It's dealing with something. What it is? Well, could be parasitic mite syndrome, might be European foul brood. It's something. We need to look at it a little further to determine exactly what it is, but that's not what we're trying to get to today. What I want you to understand is that this is not right. This is correct 100% of the time when you see your larva C-shaped at the bottom of the cell, a healthy white color. And I should mention that there should also be food in there. And you can see in all of these cells, there's that jelly at the bottom. This colony has these not C-shaped larvae. They don't even look like larvae anymore. This debris on the bottom, this is a larva that has melted, literally melted to the bottom. This is not a healthy colony. In addition, you can see that they're off color. That is key. Again, you do not have to be a professional to understand what is healthy and what is not. If you do not see what's on the right when you're looking at your brood, and you should when you do an inspection to determine disease presence, if you see something like what's on the left, you know something's wrong. And it's time to get somebody else in there. If you don't know how to determine what it is, it's time to get somebody else in there. Or better yet, before you get anybody else involved, hit the books, reputable websites. Michigan State University Apiculture is a good place to start. University of Minnesota has a great honeybee program. They're a great place to start to get resources. We all have resources on the health status of bees and how to determine what these diseases are. But your first spot as a beginner is to be able to determine what's healthy and what's not. And this is key. What I'm showing you on the left, the reason I picked this picture is because this is pretty typical of what we see. European foul brood is something we see a lot of in Michigan. American foul brood, I honestly don't think I've ever seen in Michigan. I think I've only seen it maybe once in my life in California. It's not as present as it used to be. It's not American foul brood. Granted, I don't want to say you're never going to get American foul brood. It's going to be rare if you get it. European foul brood is typically what you're going to be dealing with, or parasitic mite syndrome, which is another thing that has recently come on stage. That it's mite driven. We don't fully understand it yet. There's a lot happening in beekeeping, and so a lot of these diseases that come on, there's some that have been more well studied than others. But essentially, again, I'm getting back to you want to be able to determine what's unhealthy or what's healthy, and then from there you start investigating. All right, so I'm going to wrap up today. I think, well, no, actually, I still got the honey supers to go through. So we've got a little bit more in this lecture. Bear with me. All right, so diseases. So I just wanted to throw out a few diseases. Again, this is not a comprehensive list. This is just a talking point. Lots of different diseases out there, some that you're going to see more readily than others. My main point here is I want you to see that when things don't look right, something's not right. So this is chalk brood. Chalk brood is essentially, it's a fungal pathogen that's transmitted by spores that are consumed by the larva as food. And what happens is when this fungal pathogen gets into the larva, it essentially takes them over and turns them into this, like, it's like chalk. It's like a piece of chalk, like a white hard thing or a black hard thing. And I don't want to say that they are kind of hard. I mean, they are mummified essentially. But essentially what we're looking at here is if you were to do an inspection and you were to see these things inside of these cells and you could pull them out, that is chalk brood. Chalk brood also, the workers will remove them. And so you generally, most of the time, you find them on your bottom boards. Now, chalk brood is something that's usually associated with lots of humidity. I don't actually know if I've ever personally seen chalk brood in Michigan. We used to get it in California when I was beekeeping out there. Every so often, usually during the wet season, we would sometimes get chalk brood, but never in high amounts. I've only been able to collect a sample from one hive over my career, and that's what I have in my diagnostic collection. But it is something that does not look right. So if you were to see this, you know something is not right. Again, C-shaped, pure white, bottom of the cell, 100% of the time. That's a healthy colony. All right, you're also going to deal with a bunch of pests. We are going to talk about pests and pathogens in detail in our pest and pathogen lecture. That's why we're not spending a lot of time on them here. Again, just I want you to understand what looks right and what doesn't. So small hive beetle. So a small hive beetle, they feed on honey, pollen, and brood. They leave deposits of yeast behind. So that's kind of what you see here on the right-hand side is these rims just accumulated with this yeasty slime. The larval stage is primarily what is the damaging stage. You typically see the adult stage scurrying around. In Michigan, the southern part of Michigan, we do have small hive beetle. It is something that can be controlled, and I'm not going to talk about. There's trapping mechanisms, there's biological controls that you can use for them, but we'll get into that in the pests and pathogens lecture. But generally, not a huge problem in Michigan. I don't know if I've ever seen a colony completely succumb to small hive beetle. I gave a talk in Virginia a couple of years ago with the Virginia nature group out there. And the Virginia beekeepers, this is a big problem in a lot of southern states. Small hive beetle will actually take out colonies in some of these regions just from sheer numbers. Luckily, we have not had that problem in Michigan. This is also a good reason why you should not be importing bees into Michigan. Whether you're buying packages or nukes from out of state, sometimes problems come with them. And that's how we have global transmission of all sorts of invasive problems, is because we move things around very readily. So again, a good reason why you want to buy bees once, and that's the only time you ever buy them, is because you're not importing somebody else's problems in your apiary. But small hive beetle is something that you can see very visibly. It's a good size insect inside of a colony. Bees, a strong colony will not succumb to small hive beetle. But if you have a dinky colony that's struggling, that's full of disease and varroa mites, I could see it succumbing to small hive beetle. But again, Michigan's not really the place we have them. If you're at a state and another state, particularly the southern states, this can be a huge problem. Alabama, Louisiana, Mississippi, pretty much all along the eastern seaboard in the south. Pretty big problem. All right, so we're done with the brood chamber. These are all the things that you have to look forward to finding in a brood chamber. A lot of stuff. Honey super is much easier to go through, so we only have a few slides on honey supers, and then I'm going to cut you all loose. All right, so honey super. So we're up above the brood chamber for this. So honey's stored in frames, and the super, they're stored as uncapped. They're stored in uncapped orientations and capped orientations. And we talked about earlier that the difference between the two is capped honey has a moisture content of 18% or less. Uncapped honey is still being cured. So they're still drawing down moisture in those cells. It's why you do not harvest uncapped honey. It has a high water content and it will ferment. You only harvest capped frames. So when you're getting to harvest season, pretty easy to determine what you're going to harvest. You're going to harvest the capped stuff, not the uncapped stuff. All right, so that's the big difference. You can see the cappings here. Like I said, they lay it down in sheets, so it's not as fine as we would see with the capped brood. Going back to that again, capped brood. Look, all those nice, perfect cells on all sides, kind of tightly stuck down with individual sheets of wax. Whereas this laid is kind of a huge sheet all together. And you can see that there is no distinct definition between any of those cappings on the capped honey. All right, so I get a lot of questions about this. And this is how we're going to kind of wrap this up. I've got a couple little fun slides here at the end that we'll touch on, but this is kind of how we're going to wrap up today. So this is something I get a question a lot about. What's the difference between these two honeys and why do the bees do this? All right, so again, in no situation between these two styles of cap. So what I'm looking at here is the whiteness. You can see that this one on the left, we call dry cappings. There's this whiteness color to it. This one on the right is we call wet cappings, and there's this kind of golden color to it. The difference between the two cappings, neither one of them is bad. This is something that genetically the bees are predisposed to do. Some bees leave an air gap underneath their cappings. So they will cap honey that has a little air gap between the honey and the top of the cell. That creates a dry capping. So the honey is not in contact with the top of the cell. Some bees do wet cappings, where the the capping lays right on top of the honey. And so it's in contact with the honey, which gives you this golden look to it. The honey's right in contact. That's the difference. There's no big deal about either one. They're both good. The thing comes back to genetics. So Italian bees typically produce dry cappings. A lot of other lines of honeybees produce both. Most of the bees that I have, especially the ones that have gone through several years that I've reared queens for, that are not any particular line of queen, they're kind of what we call Michigan mutts. They'll do both. So you could have colonies that do both of them, one or the other. Don't get hung up on this. This is not a big deal, but that is one thing that we see a lot of. So essentially in these honey supers, all you're going to see is honey. Now we talked about earlier that the brood chamber just occupies those two deeps. Typically, she will not, the queen, as I'm saying she, but the queen will not go above that area of the brood chamber. If you want to ensure that she does not go above that, you can put a queen excluder on top of your brood chambers. But for the most part, she won't go much beyond that second deep or that fourth medium, depending on what you're using as far as equipment. And that's all that's going to be stored in those top supers is honey. So this is what you're going to see there. Okay, we're going to play a little game here real quick. I just want to make sure I've got all the slides I want here. Yeah, all right, great. So this is a game that you can play at home with yourself and your kids. It's a really quick, easy game to play. All right, so what is this? Y'all, we just went through inspections. We just talked again about caste and sex of bees. You have 3 bees here that have arrows pointed to them. Each one of these, what is the sex and what is the caste? So, the sex of the bees. 2 of them are females, 1 of them is a male. Which one is the male? Well, if you answered B was the male, you're correct. This one right here is a drone. Again, look at the eyes. They occupy the entire head. Even from a top view, they occupy the whole head. And you've got the furry butt on it, right? That rear end of the abdomen, that is furry. That's your drone. Your queen, look how long that abdomen is. In fact, it's hanging into a cell. That's how long it is. That's your queen. And then your worker, the smallest of all 3 of them with the small eyes. Again, the queen has the small eyes too. That's your worker. All right, so there's our 3 types of bees. You should all be experts in determining the sex and the role of each of those bees. Okay, we're going to do a little experimentation on cells here. All right, so we have 3 types of cells on this frame. We have A, which is pointed to these bigger cells down here. I know it looks like it's pointed to an open cell. I've been called out on that before. I'm not pointing to the open cell. I'm pointing to the cells below it. We have 2 big cells here, and then all of these cells right here. So, time to get the kids around. What is what? Give you a second. What are those B cells that are pointed down? What are those A cells that are really big like a Cocoa Puff? And what are those C cells that are real flat? Well, hopefully you were able to tell that those 2 top cells are queen cells. You can see that they're coming off of the face of the frame. The worker cells are those flat cells, and the drone cells are the Cocoa Puff cells, the real big ones. Because why? Well, the drones are bigger bees. All right, our last test for this lecture. I hope you're all enjoying this as much as I am. Normally, this is a fun thing to have when you have a classroom full of people. They all like to shout out answers. I hope you're shouting out answers at home. All right, so we have four things going on this frame. You all have seen this frame before. I want to know what A is. I want to know what B is. What is C? And what is D? So recalling this is a brood frame, right? So we have those three distinct layers. What were those layers? Give you a second here to think about this. Let the kiddos make their guesses. All right. Well, just like we said, in all brood frames, the general orientation is you're going to have honey on the top layer, that outer layer around the top of the frame. Within that, you're going to have a pollen layer, our power band that sits right between the honey and the brood. And then below that, we have two different types of brood here. We have open brood and sealed brood. So this is our introduction. This is the lecture that we do in addition to the gentle inspections lecture. This is what we do as 2 lectures to try and get you ready for getting into a beehive. We talked about biology. Now we've talked about what to expect to see when you get into your colony and where those things should be. And now this next lecture that you have on gentle inspections is going to be looking at how to handle the bees properly when you're going through the colony. I hope you enjoyed this lecture, and we will catch you for the next one.